Yes, I do realize that everyone seems to use the Occam's razor concept to buttress their own viewpoint. Count me in. So here we have Sasquatch. What is the simplest explanation for this species consistent with the known evidence? Many point reflexively to Gigantopithecus surviving from ancient times. One problem with that idea is the scarcity of evidence. This theory is a frail construction, given that science does not even know how this ape's body looked, whether or not it walked upright. All we possess from throughout Asia are a few teeth and jaw fragments, not enough to fill one of these. The other problem with the Gigantopithecus explanation is Sasquatch DNA, which I will get to shortly. Melba Ketchum has found that the species is a hybrid, like many examples in zoology, and that it arose from an interbreeding of human females with males of another primate species now extinct. That original male progenitor need not even have been as large as the Sasquatch that we see today. Ligers, for instance, are a cross between a tiger and a lion, and, remarkably, they are much larger and stronger than either parent. But let's slow way down. The point of this video is to lay out the case for Sasquatch as a human hybrid. As you know, the most common eyewitness description is some version of, "It looked like a human and a gorilla at the same time." To be specific, I'll first present three categories in which this creature seems to represent a blending of ape and Homo sapiens characteristics, and then I'll move on to show how Ketchum's work backs these observations up genetically. Non-human great apes have open, forward-facing noses, whereas we have what are called hooded noses, downward-facing. I won't add an image here. I think you're familiar. By all accounts, Sasquatch also have hooded noses, whereas many other features of their skull and musculature more closely resemble those of non-human great apes. A is chimpanzee, B is lowland gorilla, C mountain gorilla, and D, of course, is you and me. All non-human great apes have an abducted big toe, oriented away from the other four toes. Sasquatch toes are grouped together, just like ours. And yet, instead of an arch, they possess a mid-tarsal break, just like the other apes. Here is an artist's rendition of a mountain gorilla foot on the left and of a Sasquatch foot on the right, the latter based on hundreds of track casts. Also, the toes are longer and more flexible than ours. One observer described them as gorilla toes arranged as if they were on a human foot. Here I defer to YouTube's Thinker Thunker from his excellent video called Cracking the Bigfoot Code.
You, me, all of us humans have at least one thing in common. Our legs are longer than our arms. While chimps, apes, and most other primates, they're the exact opposite. So where does Patty fit in? To explore this further, first I needed to know if there was a normal range for human body ratios, or does it vary from person to person? So, to help illustrate human diversity at extreme levels, and lo and behold, after measuring them, I found that their arms were exactly 20% shorter than their legs. And after measuring Patty, I found that her ratios, they're not off by just a little. They're out of the ballpark off. Patty's arms are astonishingly only 5% shorter than her legs. And that puts Patty smack dab right in between humans and other primates.
At this point, you probably want me to turn to Ketchum already. But to lay the groundwork, we first need a short course in the two kinds of DNA. Nuclear DNA is what we're all familiar with, the massive combination of genetic information from both parents residing in the nucleus of each one of our cells. But there is a very important second type.
Mitochondria are structures inside the cells of the body that are responsible for energy production. They contain their own DNA, and they replicate independently from the cell. Mitochondria are passed through a mother's egg directly to both her sons and daughters. In turn, her daughter, but not her son, passes mitochondrial DNA to her children. Since it passes from generation to generation with very little change, mitochondrial DNA is a rich source of information for deep ancestry, going back thousands of years.
By sequencing an individual's mitochondrial DNA, we can tell where in the world their ancestors came from on their mother's side. These various geographical points of origin are called haplogroups.
Historically, people close to each other tended to share more genetic markers, the little DNA typos passed on from parents to their kids, than they did with people on the other side of the world. It makes sense. After all, who were they more likely to have babies with? The people living nearby. Over time, this created regional patterns of genetic variation. So the people living in, say, northern Spain had more markers in common with each other than they did with people in Indonesia. These regional patterns of variation allow us to track the migratory paths of your ancestors. By looking at where in the world your markers are found, we can figure out where you came from.
Okay, now, the astonishing breakthrough made by Ketchum and her team was to establish that Sasquatch mitochondrial DNA is human, while their nuclear DNA is not. This finding was met with an entirely predictable backlash by those of many ideological and religious stripes who are unable to entertain the possibility of humanity's mixing with another species. The knee-jerk accusation was that Ketchum's samples must be contaminated, so she made a video persuasively laying such charges to rest. We non-geneticists will find some of this discussion over our heads, but still we can tell that she knows what the hell she is talking about. Here's my challenge to skeptics out there. If you have a specific objection to any of the science that she mentions here or in the much longer version of her video linked below, explain it. Or if you can find a scientist, a geneticist, who can point out some mistake she's making, why not bring her or him into the discussion? If you have a particular doubt about any one of the samples that you know she tested, such as the Justin Smeha tissue sample, understand that the Ketchum team ran mitochondrial sequencing on 109 samples, each with a very concrete link to Sasquatch sightings.
We're going to show you how we could determine with multiple types of testing that there was no contamination in the samples. We have a little vortexer that is extremely vigorous and rapidly moving, and what you do is you put an alcohol and water combination onto the actual samples, hair in this case, and you vortex them with this professional vortexer, which will shake loose any loose cells, because that's where where you touch something you'll leave a cell which contains DNA, and this shakes loose any cells. And we did this multiple times per sample to make sure that there was absolutely no foreign DNA on the hair, that the only DNA that was there was in the actual hair. So we did this to every sample we could. Now, of course, there were some samples in the study that we couldn't wash, those being saliva samples or other liquid samples. But there's not a problem with that, because you can always tell if they're contaminated or if it's a mixed profile whenever you move forward into the actual DNA testing. Okay, now here's an actual yield gel from the study. This was using the hypervariable region 1 primers, THR and DHL, which are universal mammalian primers, and we use these commonly for species identification in forensic cases. You see the three Erickson samples, 37, 29, 31, and you can see that nice clean bands. There's no other bands around them. There are no no contamination there, strictly human. And this is in mitochondrial DNA now. And if there had been any DNA contaminating any of those reagents, or if we had handled the samples inappropriately, there would have been a band present there. This shows that there was only one species, and it was human, in the mitochondrial DNA of these Erickson samples. Of course, this was preliminary. This is before we even sequenced. Now here's the graphic representation after sequencing that the machine puts out. There's 4 nucleic acids present, and they're each designated by a different color, and that gives you your DNA sequence. You can see the abbreviations for them at the top of each of those pictures. Cytosine, adenine, thymine, guanine, the 4 nucleic acids are abbreviated by the first initial, and the order that they're in is what makes you an individual. So these are 2 different individuals, and in this short little piece of sequence, there's 6 different polymorphisms, or SNPs, or single nucleotide polymorphisms. These are mutations where the top individual is different, or a different individual than the bottom individual. And the errors point to where you have these individual differences in the sequence. Now, when you have contamination, it would be like one of these was with an overhead projector. In the old days, you could have a clear graphic and you could overlay it on top of another graphic. Well, this is what happens when you have contamination with sequence. It's very obvious. You can see it and you know you have a problem. You know you got contamination. And if it's a different species, you're going to see a ton more of these single base differences, and they're superimposed. So once again, it's easy to tell if you have contamination. This would be a contaminated sample. It's not one in the study because we didn't put any in the study that were contaminated, of course. A contaminated sample is going to have probably at least 50% or more of a different base laying on top of another base, where there is a SNP or a polymorphism or a change in sequence from one individual to the next. This is the same sample run twice to confirm. You see that there's a green peak that's a little taller than the black peak in the left-hand sample, but they're sitting at the same spot, and you know, there's some background at the bottom, but it's not nearly as tall as that one peak in reference to the other peak. The same sample on the second run, they actually came out equal on top of each other, but we didn't have any of this with what we saw. We went forward with the testing after we would get clean sequence, as it would have obviously shown. But there's other ways to look at this too, and let's look at one of the samples in the study next. Okay, this is human mitochondrial sequence obtained from sample 140 in the study. There's a little bit of background, but once again, it's way too little to have caused there's no contamination. It's just background, and you can see how clean the peaks are and clear, and you can see the different colors. It's clearly human and blasted human. Blasted Native American type, and then we did the whole genome on this one.
When she says blasted, this simply refers to the acronym for Basic Local Alignment Search Tool, a standard in forensics for identifying species using a universal database. At this point, I thought I would present a case study of just one of the more than 100 samples gathered for the study. This is in tribute to a friend to many of us, J.C. Johnson, who recently passed away. Most of the organic samples in the study were hair, but there were a few extremely important blood samples, one of which was recovered by Johnson's team.
Here's a scenario. We're out here in Upper Fruitland. This skunk is dead. The scent gland's been ripped out. In fact, I'm hardly smelling anything here right now. The scent gland's been removed. The guts have been removed. Now something rolled that irrigation pipe from about 30 yards away, and we know why it rolled the irrigation pipe out here into the field. The skunk was inside of it, and it wanted to eat the skunk. Let me show you further. That's blood inside the tube, isn't it?
Yeah, we actually took some samples, we swabbed, and we're going to send some off to get them analyzed. Basically what happened was something looks like, with the dead body over there, the skunk, it looked like the skunk may have come through this 8-inch pipe, PVC, and whatever was after it was able to punch 2 holes into it, trapping the skunk.
Yeah, like on either side. But you can see back in here, we've got blood.
We got blood back here.
Fingerprints and finger swipes where he went chasing the skunk with it. There's blood everywhere.
Yeah, it would take a lot of force to punch holes through this. For me, it would be impossible.
So he punched holes on either side, probably trapped the skunk in the middle, and he got away, went up the pipe, and he went after him and reached for him.
Yes, sir. Basically, we also got what might be teeth marks going across here and across here, or it's the fingernails. Got some up along this way also.
Yeah, look at that.
And from what Brenda was saying, this piece of PVC was, it was settled over there at first.
Yeah, so the skunk goes inside it, the big furry one rolls it out here.
Or carries it out here.
Or carries it out here to go after the skunk. And he obviously got the skunk. The weird thing is, the scent lines are removed. I shouldn't even be able to go over and film that skunk, and it's pretty tolerable. The only thing I smell is probably a spray during the struggle. That's all I smell. But we got plenty of the curly hairs that don't belong to the skunk that we're going to collect.
You know what, this makes sense because one, if this was still over there, if the skunk was still in it, it could have bolted out and gotten into the
bushes.
Got away.
That's why he brought him out here into the field.
There's nothing out here. It's just...
He had to have carried the pipe because the pipe was actually supposed to be turned around this way. So this end over here should be on that side because they hook up the pipes once they get done cutting and baling.
So the mating flange would have been on that side instead of this side.
There you go. So he actually turned the pipe around and carried it out this way.
That's ingenuity. They like skunks, guys. Yeah. About a mile from here as the crow flies, we've got peaches, pears, apples, baby corn coming up. Somebody wanted a skunk snack, but we've got blood everywhere, and we've got bloody fingers running up the pipe. You can't probably see it, but there's definitely, they're as white as my thumb.
We're on here on this pipe, so the skunk's in there, he's trying to get it, so he's ripping these off like this. That's where you get all these nails scratching in there.
And there's 5.
If, with all the young ones that have been sighted in this area this year, if I had a youngster I wanted to hide out and all the animals would leave it alone, the predators, the dogs, the coyotes, whatever, I would take skunk scent and put it all over my youngster and everything would leave it alone. A skunk lays by the side of a road until he evaporates. Ravens won't touch it, buzzards won't touch it, so skunk's a good mask, a good smell to hide behind.
Ketchum is not the only researcher over the years who has tested Sasquatch samples and found mitochondrial DNA matching human profiles. I link to one such study in the description below, and what follows is another involving a highly respected genetics lab in Denmark that extracted DNA from a hair sample recovered in Sumatra near a sighting of orang pendek, their local version of Sasquatch. Structural analysis of the hair shafts ruled out a human source, and yet the mitochondrial DNA sequence came back human. Here, once again, this result brought accusations of contamination.
I think initially what was interesting about the sample was the hair color, right? They had these short fragments and they were this sort of orangey color that's at least not common to the native people of the area.
Here's the accuser, whom we'll hear from soon. He simply cannot believe that such non-human hair could test positive for Homo sapiens without some amateurish mistake.
It's the moment of truth for the potential orang pendek hair. The DNA sequence is now available for analysis.
And we did our standard DNA analysis. We basically extracted the DNA from the hair. We had a complete identity, 100% match over the region we looked at to human DNA. This led me at the time to the conclusion that it's basically a human.
I don't think this hair sample could have come from a human in any way because the structure is all wrong. Human hairs are very characteristic. And this one does not look in any way like a human hair. The human DNA that Tom Gilbert and the DNA lab found could have come from humans. It could have been simple contamination when the hairs were sampled. Somebody forgot to put gloves on or just loosed a drop of sweat on the hairs.
So our American study locates itself on an already well-trodden path, always the target of detractors with a rough and ready allegation that then fails to hold up on scrutiny. Unfortunately, Ketchum handicapped herself right out of the gate, making a dreadfully hard sell all that much harder. The journal Nature did not accept the study, so in frustration she finally released it herself, which violates scientific protocol. She has a very user-unfriendly website. And she held a truly disastrous 2013 press conference. Add to these the fact that she's a woman, a blonde southerner no less, and a veterinarian rather than an academic, all of which still face an unjustified level of disdain within the mainstream scientific community. And finally, Ketchum is a born-again Christian, a fact that would sadly raise suspicion in the minds of many, even if she didn't, furthermore, occasionally mention that Sasquatch may be the Nephilim of the Bible. None of these factors, however, bears upon her area of career expertise and the capacity of her team of forensic specialists, all with PhDs and/or decades of experience in the field, to perform the extremely routine, straightforward task of sequencing and interpreting mitochondrial DNA contained in these sample cells gathered across North America, and to assign them to various human haplogroups. The immense difficulty only arises in the question of how to decipher the nuclear DNA, which is vastly larger and more complex, and, in this case, utterly unprecedented. The Sasquatch genome has never been sequenced before, and therefore no species database on Earth holds a match. There is nothing to compare the findings to, and nobody is qualified to read the sequences the team found, including the team itself. Until we learn about the male progenitor of the Sasquatch species, the genome is a black box. Here's the bottom line, though. The very fact that the mitochondrial DNA came up human, while the nuclear DNA of the very same organic samples clearly did not, is Melba Ketchum's lasting triumph, and will find its place in history. This bizarre genetic discrepancy is enough to show that the organism under consideration emerged from an era of crossbreeding between female Homo sapiens and males of another species, as yet undiscovered. I'm baffled not by the resistance of mainstream science, but by that of those of us who know of Sasquatch. There is some willful denial at play here, some refusal to put two and two together. People seem to want to push their own agendas, or would rather wallow in the misty mystery of it all. Anyone who wishes to successfully attack Ketchum's fundamental discovery here will need to find fault with her raw mitochondrial DNA data, and none of her critics across the years has yet met this challenge. In the 6 years since Ketchum released her findings, we have seen an explosion of new discovery about the central role of hybridization in our own evolution. This serves to make much more plausible now the claim that Sasquatch arose through hybridization, and is therefore our next of kin here on Earth. Occam's razor dictates that we accept the simplest explanation consistent with the evidence. Why are there no persisting hybrids of humans with Denisovans, Neanderthals, or anyone else? Well, there are, of course. And this truth is as plain as the nose on your face.